Okay, so for part two of this, this is the science of the neurobiology of what we currently know. I've been doing a lot of research, a lot of reading on this. This is fascinating to me because for many years I understood the theories and I worked in the classroom and I said this is not enough because there's a lot more to children. And so some of this stuff has been around for about 20 years, other stuff 15 years, other stuff 10 years. So let's go through this. And we'll start right here. And this is the child. This is a zoomed close-up of a child's brain. So when a child is born, we'll talk about this again for infancy, but a child is born with just about as many neurons as they will have as an adult. It's about 80 to 100 million neurons. All right, and so what does that look like? I like to say, if we put it metaphorically into a grain of sand, that would be about a truckload of sand, just for neurons. Now, each one of those neurons, after a child doesn't have any kind of neural connections connected, well, they don't have that many neural connections. You can see right here, these neurons are not connected with one another. So as the child's learning, now we go back to Piaget's work, a child is experiencing the world. They're having conversations with, well, not necessarily having conversations, but adults are talking to them. And what happens is these neurons develop synaptic connections. Each neuron has about 1,000 synaptic connections, and there's a synaptic connection that connects to another neuron's dendrite or dendritic connection. And that synapse sends neurotransmitters to the dendrite, which then tells that neurotransmitter what it's supposed to do. These memories get stored in here, so it fires this way and that way. These become more efficient with more experience. So what happens is, with more experience, not only are there, now science has confirmed that new connections get made, but also myelin, which is a sheath of fat, goes and it wraps itself around each neural connection, which then allows the signal from one neuron to another neuron to go quicker. And so these move, I can't even tell you the speed that they move at, but you just have to think we have these neural connections, these neural networks that allow us to do everything that we're doing. So at birth, we don't have those neural connections. By 6 years old and by 3 years old, we have far more neural connections that we will ever need. So what happens is all this learning, all this input, all these neural connections then get pruned. And then pruned is when all the neural connections that we don't need, we don't practice, we don't do these things, they start to fade away. And it allows for the neural connections to become stronger and these neural networks to take shape, and for us to then become the adults, the abstract thinking adults that we become. So even looking at the 14-year-old, the brain, we still have the same amount of neurons. We've got this 80 to 100 million neurons. However, we don't have the same level of synaptic connections. Now we have these synaptic connections, and that was something that has been known for quite some time. Now it wasn't known exactly how they connected with one another, how learning took place. That's for another conversation that we'll have as we move through this course. What they started to discover right around in the 2000s was an idea of epigenetics. So what happens is we have 23 chromosomes that come from the mother, and 23, or the woman, and 23 that come from the man, the female, the male. They provide this, and those come together to create 23 pair. So we've got 23 pair of chromosomes in every single cell, and every single cell has the exact same DNA, but different protein or different acids, amino acids that make up those DNA, which are our genes, the combination. Certain ones get turned on and get turned off to say that this gene or this cell is going to be a blood cell versus this cell that's going to be a neuron versus this cell that's going to be a white blood cell or a skin cell or whatever it is. It's the exact same genes, exact same, but it's different genes that have been turned on and turned off. Now, the fascinating thing that emerged from research about 20 years ago was the fact that our experiences have an enormous impact on what genes are turned on, what genes are turned off. Here's a picture of that neuron. So here are the synapse, the neuron. Here are the axons right here that send neurotransmitters to the dendrites. So we have about 1,000 of these branches coming out for axon, connecting to thousands of then neurons. Here's 1,000, so it creates this tight network. And what will happen then is the DNA will ultimately change. You can look at this graphic a little bit more, pause the video, look at it. These regulator proteins are what grip on. They're created by stress, by experience, not necessarily just stress, but we think about how children change and those changes in those twin studies that we looked at. It looks like, there are a lot of things that are the same, but there are a lot of things that are different. And those things that are different were from the environment. And so the environment provides these changes, these influences, these experiences that will then alter the DNA, and then that DNA gets stored and becomes part of the genes that we hand down to our children. And so we can even start talking about these generational issues, the impact of issues that were, we can say slavery, we can say great migrations, we can say these other things that had a huge social impact on individuals that changed the genes, that turned them off, turned them on. This is all still research in action. Okay, so now what are the things? Here's a list of what creates those gene regulators. We've got positive things, we've got things that aren't so positive. So just one-to-one interactions with adults, healthy family relation, healthy healthcare, or solid, reliable, dependable, functional healthcare, playing with peers, self-directed exploration, hands-on learning, a family with financial stability, comfort, good nutrition, exercise. All of these provide gene regulators that turn the genes on and turn the genes off. Now this brings in the conversation of a lot of the issues that we consider genetic, but a child has, or a person is genetically predisposed. So that doesn't necessarily mean they're going to develop Alzheimer's, they're going to develop alcoholism, they're going to develop autism. It means that they're predisposed, and that predisposition is based on environmental experiences. For example, an individual who is predisposed to alcoholism isn't going to have those gene regulators that turn on that addictive gene until they start drinking alcohol. Now, the things on the negative side are poverty, caregiver mental illness, maltreatment, single parent, exposure to violence, economic hardship, low maternal education, in-home substance abuse, parents' incarceration. These are all those ACEs that we talked about in class, the adverse childhood experiences. And what these do is it turns on the genes that then send neurotransmitters to certain regions of the brain and activate certain regions of the brain that shouldn't be activated at other times. We talked about what Nadine Burke-Harris talked about in her TED Talk was the amygdala. So our amygdala fires when we have this fight or flight. And they say it's more than that, but we've got our fight or flight response more or less in the amygdala. And what happens when we encounter a bear or something fearful is neurotransmitters send this dopamine straight to the amygdala, and the amygdala fires all these other messages out to the cortex and says, this is what we need to do. And it doesn't go through these other midbrain areas. It just goes straight out to the cortex. And the cortex, there's no processing, there's no filter. And so we do things sometimes that it wouldn't even be, if we had the time to step back and think about it, we might not do it. This is just an immediate response, almost like it's reflexive is really what it is. And so what happens is when children are consistently exposed to these adverse childhood experiences, the amygdala is always firing, and the amygdala becomes larger. And so children and people who have this larger amygdala are more likely to have that message, just default message, go from the amygdala to the cortex. and there isn't any kind of filter from all the other regions of the brain, and that creates a lot of challenges. Now this graph wasn't provided to us, but when we have ACEs, these are the risks for developmental delay. So a child with 1 to 2 ACEs, they have a higher risk than a child who doesn't. Now we get up to children though who, and we talked about this last night, children who have 4 or 5 or 6, we get a child who has 7 of those ACEs, they are going to have a developmental delay. So this is something that we need to consider. However, the challenge that we face is, well, what do we do with that? And how do we respond to that? And what kind of therapy and intervention do we provide? That brings us to some other conversations we will talk about, but we can talk about attachments. This will bring us back to theories. Attachment, that secure, insecure conflict that Erickson talks about. So there's a lot that goes into it still. And the brain will change. So this is a graph that shows us the amount of stimuli that is required to impact that change. So you can see clearly that you need a lot less stimuli when a child is young. But as a child, and you can really change the brain. So the child's brain is most influenced in these first 10 years. It is so significant. And then as they get older, the experience doesn't have as much of an impact. Now, on the other hand, the effort to change it doesn't take as much intervention to change that in the first couple of years. But as we age, that becomes much more difficult. So we get at this 30 years old, and this is really that marker where children and people before 30 years old are significantly higher to have maladaptive behaviors, incarceration, substance abuse. And on the other side, we have, people who are leading social civil wars, for better or for worse. They're changing the world through climate change, these demonstrations, these battles, and it can be very powerful at that point in time. So it's for good or bad. Once we get to 30, a lot of that stuff is already set in place. Okay, so what does this bring us back to that gorilla study where I said, okay, what does it have to do with it? And the thing is that we have the brain science, the neuroscience. We have this theory. The challenge is that we still don't understand the individual child. So we get caught all the time looking at the ball. The ball represents the theory and the science. Now we've got it. The problem is that what does this really mean to each individual child? And so that's something that really the ACEs research has started to uncover, but there's a lot of research that I've been doing with some colleagues of mine that reveals some information that allows us more to contextualize this to the classroom and the complexities that it relates to in the classroom. And what it's referred to is positionality. So a theory of positionality. And so what it starts out with is we have this habitus. A habitus would be the classroom. It's the environment. It's the microsystem. Then we've got the positionality. So the positionality is us within that habitus, us within that classroom or microsystem. In there are so many components that make us who we are, a unique individual. Who is this child? So to start to conceptualize this, there's our social identity. That's our race, our gender, our ability. These are things that are socially constructed. If I am in certain areas, in a room full of white people, then my race is not going to make much of a difference. If I am in a room where everybody it doesn't have a visual disability, we're just going to assume that disability is not an issue. These are all things that have a meaning because of the social content, the social location that we're in, the habitus. Then there's the self. The self is really what we think about for ourselves. Groups and communities really inform what the self is. We identify ourselves because of the groups that we're part of. We become part of those groups because of our social identity often. And then there's that biological piece to it. So when we talk about disabilities, we talk about abilities. A lot of this goes to those ACEs. How does our body really respond? How are we feeling in that situation? And then last, we have our cultural values, our morals, our roles, our understanding of rules, customs, symbols in our culture, and all this works together to create each person's social positionality in a classroom, in any one-to-one conversation. And so that's the end of science. We'll continue to talk about all this as we move through the semester. Thanks.